Learning with Illuminati. Part 17. Introduction to the Cerebellum. The cerebellum, alongside with the basal ganglia, are important for regulation and controlation of movements. The cerebellum modifies the activity of upper motor neurons by detecting and reducing differences between intended and actual movements, and do also contribute to cognitive functions such as working memory, reasoning, task flexibility, language, and problem solving. In a nutshell, the proprioception of the body goes via the spinal cord to the cerebellum, parallel with intentional information from the cortex, via the pons. The cerebellum compares this information, and calculates suitable sequences for future muscle contractions needed to achieve the actions. The output from the deep cerebellar nuclei is relayed via the ventral lateral nucleus of the thalamus to the primary and premotor cortex. Problems in this circuit may cause ataxia. Imagine that you unroll the cerebellum. Some basic divisions of the cerebellum are the spinocerebellum, which receives input from the spinal cord; the cerebrocerebellum, which receives input from cerebral cortex; and the vestibulocerebellum, which receives input from vestibular nuclei in the brainstem. The vermis, or the median part of the spinocerebellum, regulates proximal muscles and eye movements, whereas the lateral part of the spinocerebellum regulates distal muscles. The cerebrocerebellum regulates highly skilled movements and speech, and the vestibulocerebellum regulates movements underlying posture and balance. Further, the cerebellum contain multiple somatotopic maps of the body, and the spinal and vestibular inputs remain ipsilateral as they enter the cerebellum. In other words, the right hemisphere receives input from the right side of the body. However, the projections crosses side when traveling between the cortex. From lateral to medial, the four deep cerebellar nuclei are the dentate, emboliform, globose, and fastigial. However, in humans, the emboliform and globose is fused, creating the interposed nucleus. Pathways to and from the cerebellum. The superior cerebellar peduncle is mainly an efferent output pathway to the motor cortex, via the thalamus. The middle cerebellar peduncle is an afferent pathway from the motor cortex to the cerebellum, via the pontine nuclei. And the inferior cerebellar peduncle has both efferents, from the vestibular nuclei and spinal cord, as efferents to vestibular nuclei and reticular formation. Also, the inferior cerebellar peduncle receive afferent modulatory inputs from various areas via the red nucleus and the inferior olive. Some efferent projections from the cerebellum are the Cerebrocerebellar, which goes via the dentate nucleus to premotor cortex, important for planning; the spinocerebellar, which goes via the interposed and fastigial nuclei to upper motor neurons, important for motor execution; and the vestibulocerebellar, via the vestibular nuclei to lower motor neurons in spinal cord and brainstem, important for balance and vestibulo-ocular regulation. Besides being important for motor regions of the brain, the cerebellum may also return information to non-motor areas of the cerebral cortex. The cerebellar cortex can be divided into three layers. These are the molecular layer, the purkinje cell layer, and the granule cell layer. The molecular layer contains stellate cells and cell bodies of basket cells. Climbing fiber inputs from the inferior olive, parallel T-shaped fibers from granule cells, and dendritic trees of purkinje cells. The purkinje cell layer contains, as the name implies, cell bodies of purkinje cells. These have their dendrites in the molecular layer, and their axons in the granule cell layer. The granular cell layer has granular cells, which receives mossy fiber input from pontine nuclei and brainstem. The granular cells and climbing fibers are excitatory inputs. The Purkinje cells are inhibitory outputs, and Golgi cells, basket cells and stellate cells are inhibitory modulatory cells. The mossy fibers from the pontine nuclei and brainstem synapses with granule cells The granular cells form T-shaped parallel fibers that synapses on Purkinje cell dendrites. Climbing fibers originating from the inferior olive modulates the Purkinje dendrites, and Golgi cells modulates granular cells, whereas stellate and basket cells modulate Purkinje cells. Of the about 10 million Purkinje cells, each one can receive input from approximately 200,000 parallel fibers, and each parallel fiber can contact up to 10,000 Purkinje cells. However, each of the climbing fibers do only form synapses with one Purkinje cell, however with up to 1,000 synapses on each, which provides a powerful excitatory drive. The Purkinje cells sends inhibitory signals to deep cerebellar nuclear cells. This inhibition prevents the output via thalamus to the motor cortex, but changes as inputs from local circuit neurons affects this activity. The mossy fibers as well as the climbing fibers brings input from various parts of the body, such as cortex, spinal cord, vestibular system, red nucleus, superior colliculus and reticular formation. Both the mossy fibers and climbing fibers synapses by shortcut directly to the deep cerebellar nuclear cells, affecting its output. Also, activity of the granule cells simultaneous with activity of climbing fibers may cause long-term depression between the Purkinje cell and parallel fibers of the granular cell, or in other words reducing the influence by the parallel fibers. A postulate goes that climbing fibers provide a teaching signal, reflecting the motor performance failure, which weakens the connection that might have contributed to a failed action, and believed to be important for motor learning. And lastly, the Golgi, stellate, and basket cells do also contribute to modulation of these circuits.